Bismillahirrahmanirrahim. The second messenger mechanism that mediate the intracellular hormone functions are described in this video. The primary messenger is the hormone itself, while the second messenger is an intracellular molecule that amplifies the hormone's signal and initiate physiological responses inside the cell. The hormone binds to a specific membrane receptor but do not enter the cell. Instead, the hormone, after binding with the receptor activates intracellular signaling pathway that generate second messenger, which amplify the signal, activate enzymes or ion channels, or trigger transcription or other cellular events. The second messenger for mediating the intracellular hormonal function are cyclic AMP, that is activated by G stimulatory protein that lead to uh, formation of adenylyl cyclase, and this activate protein kinase A and phosphorylates enzymes. Another second messenger system is calcium ion and other calmodulin, and this calcium calmodulin system increases intracellular calcium concentration that activate calmodulin dependent kinases. Then there are products of membrane phospholipid breakdown, diacylglycerol and inositol triphosphate pathway, and they are activated by GQ protein that activates the phospholipase C. And the major effect is that the inositol triphosphate increases calcium concentration, while diacylglycerol activates protein kinase C. The hormone that use adenylyl cyclase, cyclic AMP second messenger system, they are adrenocorticotropic hormone, angiotensin 2, calcitonin, catecholamines, corticotropin releasing hormone, follicle stimulating hormone, glucagon, growth hormone releasing hormone, human chorionic gonadotropin, luteinizing hormone, parathyroid hormone, secretin, somatostatin, thyroid stimulating hormone, and vasopressin. So the hormone binds with the transmembrane receptor, and this transmembrane receptor will and hormone complex will result in coupling of receptor with the G protein. If this protein is G stimulatory protein, there will be activation of enzyme adenylyl cyclase, and this enzyme adenylyl cyclase activation occurs at the intracellular end, and this causes the conversion of small amount of intracellular ATP is converted into cyclic AMP. The activation of the cyclic AMP or formation of the cyclic AMP will result in activation of cyclic AMP dependent protein kinases, and this will result in the phosphorylation of specific cell protein, and finally there is the effect of the hormone or cellular activity. The effect of the cyclic AMP is that it activates plenty of enzyme. There is first activation of one enzyme that activates another enzyme that activates a third one, and many enzymes or chain of enzyme or cascade of the enzymes are activated by the initial cyclic AMP formation. So this is the mechanism by which few molecules are responsible for an amplified response. So the response is dependent on the binding with the specific protein or the activation of the cellular machinery. So if the cyclic AMP binds with the inhibitory G protein, there will be blockage or decreased activity of the cell. And if it binds with the G stimulatory protein, then there will be activation or increased response of the cell. So this activation will result in uh, many functions such as muscle contraction and relaxation. Various kinases and proteins are formed that are finally responsible for the contraction of the muscle or relaxation of the muscle. They are responsible for the secretion from the cell uh, when these adenylyl cyclase uh, activation result in cyclic AMP formation. It acts on thyrotroph or thyroid cuboidal cell. They will result in the formation of T3 and T4 hormone. The same mechanism activation will result in adrenal hormone when the adrenal cell is affected through this procedure. Then they are also responsible for altered permeability, as the activation of cyclic AMP will result in increased water permeability from the distal and collecting renal tubules. This diagram is illustrating the cyclic AMP second messenger system used by a G protein coupled receptor to transmit hormone signal into a cell, triggering a cellular response. So how this occur? This is a hormone. This red dot is a hormone that binds with a 7-pass receptor, that is the transmembrane receptor, and after the binding with the hormone there is conformational change in this uh, receptor, and the alpha, beta and gamma trimeric protein that were attached to this interior of this interior side of this receptor are detached, and then this alpha subunit, trimeric protein subunit, when it is detached, it binds with the adenylyl cyclase. This adenylyl cyclase, this alpha subunit, this exchanges GDP for GTP and become active and detached from beta and gamma. Okay, and then the activation of this adenylyl cyclase occur. The activated alpha unit interacts with this adenylyl cyclase, and this stimulate adenylyl cyclase to convert ATP into cyclic AMP, and now cyclic AMP formed. This cyclic AMP activates cyclic AMP dependent protein kinases that are also termed as that protein kinase A. The active form of these kinases, uh, they phosphorylate specific proteins using ATP and turning them into protein phosphate or phosphorylated proteins. ATP and protein, they are converted into phosphorylated protein or protein phosphate, and also this ATP is converted into ADP. So finally, after this phosphorylation of a protein or cascade of the protein, these phosphorylated protein, they trigger a specific cell response depending on the type of the cell and the proteins that are involved. So here this image is presenting the signal transduction pathway where a hormone acts on a G protein coupled receptor and trigger a cascade through cyclic AMP and alter protein activity and ultimately change the behavior of the cell. So there is another mechanism, that is the cell membrane phospholipid second messenger system. Phospholipid second messenger system. So here the angiotensin 2 catecholamines, gonadotropin releasing hormone, growth hormone releasing hormone, parathyroid hormone, oxytocin, thyrotropin releasing hormone, and vasopressin, they they are responsible for activation of transmembrane receptor, and then transmembrane receptor is responsible uh, for the activation of the phospholipase C, that is attached to the inside projections of the receptors, and this enzyme catalyzes the breakdown of the phospholipid uh, of the phospholipid in the cell membrane, and the PIP2 or phosphoinositol biphosphate, or it is divided into diacylglycerol and inositol triphosphate. And this inositol triphosphate mobilizes calcium from mitochondria and then endoplasmic reticulum. And calcium ions are responsible for their own second messenger effects, such as smooth muscle contraction and changes in the secretions of the cell, while diacylglycerol activates protein kinase C, and this protein kinase C is responsible for phosphorylation of large number of proteins that exert the response of the cell. The lipid portion of diacylglycerol is arachidonic acid in nature, and this is the precursor of prostaglandin and various other uh, local products or local hormones that causes multiple effect in the tissue. So the for the products of prostaglandin, you can refer to the inflammation playlist, where these prostaglandin and their arachidonic acid, prostaglandin, and various derivatives of arachidonic acid are described. This picture illustrates the phospholipase C diacyl glycerol second messenger pathway, that is a key signal transduction pathway used by peptide hormone to trigger cellular response via G protein coupled receptors. So whenever a peptide hormone binds to a receptor on the cell membrane, that is, this hormone is originating from the extracellular fluid, and this receptor activates an associated G protein that is located on the intracellular site of the membrane. Activation of the phospholipase C then occurs, and this activation will the result phosphol result in the degradation of phosphatidyl inositol 4,5 bisphosphate that is cleaved into inositol triphosphate and diacylglycerol. This diacylglycerol then it is responsible for phosphorylation of protein kinase C, and this protein kinase C is then responsible for the phosphorylation of the one protein or cascade of the protein, and finally there is a cellular response. Then the inositol triphosphate is another second messenger system through this same hormone, and here this inositol triphosphate releases the calcium ions or calcium from the endoplasmic reticulum, and this release of calcium from endoplasmic reticulum and from the mitochondria will result in increased cellular concentration of the calcium ion that will result in response of the cell. So the same result can be achieved by a drug. Here you can see a drug binds to a receptor and then stimulates the G alpha, and this will result in activation of the phospholipase C that is responsible for these two different pathway activation, and phosphoinositol triphosphate and diacylglycerol. So increased calcium ion concentration will result in within these parietal cells, they will result in release of the gastric acid secretion. In the neuron, they will result in depolarization of autonomic and CNS neuron, and in the blood vessels, they will result in vasodilation. Thank you for watching this video. If you have any query, please write in the comment box.